what is the challenge? I mean, medicine's pretty good. You know, you go to casualty, or you go see a doctor, you get sent to get medicines, you don't suffer, you dilate, you know. You might be a bit dependent for the last 10 years, particularly if you're getting to retirement age like me. You know, you become demented, and then for 10, 15 years, I depend on you and working, paying your taxes so that I can have my social security. So everything is in order, right? Everything's perfect. But the real problem is that neurology is an old discipline. It's 150 years old. It started with this geezer called Broca in Paris, who put together a man who couldn't speak, with his brain after death with a big hole here. And he said, oh, structure and function go together. He presented his results the next day, by the way, at the Society, Parisian Society of Pathology. So that's what you need to do if you want to publish next day. Manuscript next day, supervisor looks at it in the evening, and then following morning it's off to the publishers. So what this means is that what we rely on for all our medical research is phenomenology. Symptoms and signs. Now, along we came with the Human Genome Project. This was a sort of grandfather of ours. And the Human Genome Project showed that one mutation can present with a number of phenomenologies, a number of syndromes. So in Huntington's disease, that's very well described. And then on the other hand, people have found one syndrome, spinocerebellar degeneration, and at the moment there are only 25 different mutations which cause the same syndrome. So what does this mean? It means we're in the land of complexity, which is your land, because you've been brought up in complexity. I was brought up in Grecian linear logic. Cause, effect, cause, effect, cause, effect, cause, effect. And of course the great change in my professional life was when we started using the Latin square design in analysing brain images. Cause, effect in this context, cause, effect in that context. What are the differences? And that's where you get the nonlinearity. Okay. So we have a very difficult situation in medical research, because we're dealing with data which is not going to give us the answers we want. The questions we pose are sometimes much more complex than we think. So for example, you get a pharmacologist and you say, look, I want a treatment for Alzheimer's disease. Right, so it's not that difficult these days to go to the literature, and what you find is that 20% of people over the age of 80 have Alzheimer's disease. That's fine. That's no problem. Except that you then go to the post-mortem examinations and you find you're wrong in 40 to 30% of cases. So your pharmacologist is constructing a drug trial because he's got an idea in his head. So he's got the group which he's going to analyse, which he's interested in, has got a 30% minimum error in it. Okay, but there's the controls, so that'll be fine. So you trot off, you say, well, I'll take you people, because you're the same age, same education, background, same sex distribution, you're going to be my normal controls. And then a pathologist stands up and he says, I'm sorry, but of the people who die normal, without the phenomenology, probably about 15, 20, possibly as high as 30% have changes of Alzheimer's disease already. And that just speaks to the fact that the brain is a very redundant organ, and that it can compensate for a lot of damage. Let me give you one simple example, Parkinson's disease. In Parkinson's disease, the first symptoms you get, the first thing that arises, is after 70% of your nigrostriatal neuronal population is dead. So that's the reserve. So this is lovely, because it means, you know, I can have my little Parkinson's disease but not develop Parkinson's till I'm 90. But it's really tough on the pharmacologist, who's sitting there creating his control group, which also has a 30% error. So now you've got 30% error variance, 30% error variance, unknown, so you've got to increase your N. And if you increase your N to 3,000 or 10,000 or 100,000, the money goes up from 10 million to 30 million to 100 million to 1 billion. And that's just because you've got a candidate drug where you don't know what the target is, because you don't know what the cause of Alzheimer's disease is. Right? Right, so how can you figure out the target? All you can do is treat symptoms and hope that you're on the right track. So we really are in a mess. So can we diagnose it? No. Do symptoms matter? Only a little. What's the way to pathology? It's always end stage. So it never tells you about that beginning where you've got changes but you're compensating for them. That's clearly where treatment should start. We do compensate. Can we define that? Do the treatments work? What do we treat? What about prevent? You know, it's a mess. And yet, something as simple as dementia is a bigger social, medical, financial problem by orders of magnitude than HIV ever was. Now, you probably didn't live through the HIV when HIV came up, when we had the 10 years when we were finding causes, finding treatments and so on, but I tell you, it was a disaster, pretty much worldwide, at least in its later phases. This is going to be a much bigger disaster. Let me just give you one example just to hammer this home. Up there you see FTD-BV. That's a syndrome, frontotemporal dementia of the behavioural variant. So these are people who are really thinking about dementia, seeing a lot of it. They think they found a nice little group. And there are 32 of them there. And Andrew Kertész from Canada is following them up, doing nothing to them, following them up over 10 years. And what does he find? He finds that 2 of them get motor neuron disease, which is a disease of wasting of muscles, like Hawking has. And indeed, they have motor neuron disease pathology. So that's fine. At the top there, he has 6, where it says no second syndrome. And of those 6, there are 1, 2, 3, 4, 5, 6 different pathologies, of which one says it's normal. Now, let me tell you a little secret. It's normal because pathologists, when they examine the brain, they just look at a little piece of it called the hippocampus. And they're pretending they're looking at the whole thing. Right? So there's no correspondence with the end pathology and the syndrome. But the really remarkable thing is 24 of them get another syndrome, of which there are 3 types. There's semantic dementia, progressive dementia, and corticobasal degeneration. Doesn't matter what it means, it's different syndromes. And with that second syndrome, there's another 12 who get a third syndrome. So depending on when you as a doctor see the patient, you'll make a different diagnosis. And then not much correspondence with end-stage pathology. So we're in a real mess. Now, what have we got to help us with the real mess? We have plenty of papers. Clinical, basic, all neuroscience. 100,000 a year in 2012, 150,000 in 2015. Masses of information, Nobel Prizes a go-go, lots of individual pieces of absolute brilliance, watching a calcium ion going through an ion channel, working out how the protein is distributed in a cell, different contexts and so on. But actually, across the scales of organisation, no theory, no blueprint, no understanding. We know, how base pairs become DNA. We know how DNA is to RNA. We know how RNA produces the proteins. We know a little bit about distribution of some of the proteins, ion channels across along axons and so on. But other than that, so we need to get all this data together. We need to integrate it. We need somehow to understand the causal relationships. Why? Well, because if you go to someone who knows nothing and say, we're going to try and discover how the brain functions and is organised, they say, don't be silly, this is a structure which has got as many neurons in it as there are bodies in the universe, it's much too complex a problem, infinite degrees of freedom. So you, of course, from now on will be able to say, I think you're wrong there. Actually, organic matter is made of 4 base pairs. That decreases the degrees of freedom rather substantially. And then at each level of organisation, if you can work out the rules, like DNA to mRNA, you can't have any old protein, you're going to have proteins which are determined by that particular pattern. And then the next level, by that particular pattern. And as you go from bottom up, simulating, so at each level your degrees of freedom go together. So actually finding the biological basis of cognitive functions may well be the easiest part of this whole business. because you simply can't do it any which way. It'll have to be done as a function of what the rules are in the architecture of the thing. So, let's go to the medical informatics thing now. We've got the human genome. That's told us the building blocks of organic matter, but it's also told us this is not a simple one-to-one mapping. Got a lot of modern neuroscience, which is fragmented and atheoretic. A lot of clinical neuroscience, which means that in the human brain we can do things down to 0.7 of a millimeter cube, centimeter cube, of a millimeter cube, 0.7 of a millimeter cube. We can measure tissue properties in an anatomical sense. We could do that quantitatively now. We can measure function, still in a relative sense. We can measure function very fast with EEG, with MEG. We have lots and lots of different ways of looking at the human brain, both normal and abnormal now, which we didn't have, certainly when I started. So I worked on the second ever CT scan in the world at Queen Square in 1974. I've worked on the first MR scanner in the world, but only fleetingly for about an hour or so, and did all the toxicology. And I'm now working with the best scanner in the world here at Lausanne, because of course it hasn't been Lausanne for the scanner. And then we have modern information technology, and that is just spiraling. I mean, you're all contributing, your profs have contributed to that. It's just amazing. The supercomputers at the moment 10 to the 16 calculations per second. And IBM, I think, 2019 roadmap, exascale, 10 to the 18. Okay, so we can do the calculations to do the modeling. It's just a question of getting it all together. HBP has got the 4 largest European supercomputers linked up by massive what's the word I'm looking for? Senior moment. What? Fibre optic, that's the word I meant for. Fibre optic cables, all ready and prepared for working with the HBP. And then there's a whole new area of mathematics, which is now possible because the computers can do the validation of the mathematics. And by mathematics I include statistics, of course, and machine learning is a particular example. So the armamentarium is all there, from moving from syndromic diagnosis, which is what we started off with Mr. Broca, to 150 years later, to now a signature, multivariate signature, which is composed of all the stuff that we know about phenomenology, plus all the images, all the EEGs, all the genetics, all the proteomics, everything we do on our patients, and which we store away in computers so that lawyers can earn a living. Now that sounds silly, doesn't it? But it's not, because every hospital has a computer and a data store. Actually, it has a number of data stores, because every department usually sets up its own little data store. And they have different files, and they have different this, and they have different that. And all that stays there for follow-up of patients, which is only about 5%. Most people go once and then go away again. And the rest of the time just sits there. All that information. Do you understand the wealth that there is there? The wealth of information, completely untapped because of things such as security, anonymity. You can't guarantee security and anonymity. Well, this is where we're going to unlock the value of the clinical records and data. We're going to go in there and we're going to Google them. Now that's a metaphor. Because you do this all the time. Those of you, no one has the computer. Oh yes, Lester does. Lester's doing something else, he said in my talk before. And it's a new talk, Lester. You do it all the time. You look for information everywhere, and it's there, in real time. Why can't we do that specifically for the niche of hospital data, and for that matter, research data, and for that matter, pharmaceutical company data on the placebo arms of failed trials? Can you imagine how much structured data there is out there which is totally useless and just sits on computers and on databases for nothing? We're going to catalyse a major collaboration of hospitals. This is what you're going to do. You're going to federate and integrate that knowledge and put it into a framework where it can be got into and where it can be used. So that's the plan. Now, what are the problems in terms of medicine? So what we're doing is reclassifying disease, right? That's one of the major things we're doing. for the disease signatures, and we're going to do that in real time, continuously, iteratively, with data mining. And that's something you're going to start tomorrow. And the history books will say that it started in Lausanne. What's today's date? 16th. On the 17th. On the 17th of March 2015. The other things we're going to do is we want to visualize that data, you know, show graphs, show distributions, do public health stuff, epidemiology, health services, and we want to also, depending on what disease signatures we find, we're going to have to go back to medics and say, well, what does this group of people that biologically and phenomenologically resemble each other, what makes them similar? What makes them different to this group, which is only different by, say, one haplotype, or by one this, or by one that? So there'll be a lot of hypothesis testing, and because the diagnoses will be more accurate and more reproducible, good evening, we'll also be able to get better clinical trials, perhaps with only 10, 15, 20 people on either side. Perhaps everyone going to a hospital will become a member of a clinical trial as soon as they start taking a tablet. So all that should be great fun. And this is just to show you, this is Ching's work. I'm not going to point to Ching because people need to find out who you are. But essentially this is ADNI AD, and ADNI healthy controls, compared to AD on the same data using machine learning and healthy controls using machine learning. Can you see the differences? It's not fantastic, is it? 75% sensitivity, 85% specificity. It's still better than the clinician who's down at 65%, 70% sensitivity. Sorry, 75% specificity. Sensitivity we don't even know. So, the data you're going to use, research and clinical, the challenge you have is to use hospital data because there's so much of it. And that's not complete, it's not structured, it's not standardized, it's not clean, it's protected for privacy, it's protected against corruption. So the Human Brain Project, the MIP, the Medical Informatics Platform, has dealt with all of those, and I'll show you in a moment how, in its strategy, but now it has to be implemented. These research databases are protected culturally. That means researchers, for the most part, are a bit silly about their data. They think it belongs to them and that it's going to win them the Nobel Prize. No one has ever won a Nobel Prize with data. They have to think about the data and get some results to get the Nobel Prize. So you can share data, and people are beginning to share data a lot, and there are agencies like the NIH and so on that are saying, no way, you get no money if you don't share your data. The drug companies are being forced in Europe to share their data now. But the drug companies are also beginning to give us data. So Sanofi Aventis have given us 400 data sets from a placebo arm of a failed drug trial in Alzheimer's disease. And they're very structured. I mean, they've got lots and lots of highly structured data. But the hospital database is where it's at. And what are the challenges? The challenges are privacy. So in the first instance, we are never going to move the data from the hospitals. We're going to move aggregated results from the hospitals with variances. And when we have a lot of hospitals, we're going to bring them together in a unified portal, and we will aggregate the aggregates. So there's no way you can go back to the individual in the first instance. If that is shown to do something, then people will be prepared, undoubtedly, to see individual data being looked at as well. And it'll start with the orphan diseases, it'll start with the fatal diseases, it'll start with the diseases that lead to long-term invalidity, and there the balance between the rights to health and the right to privacy, society will decide on. So the critical thing is to get that first bit right. So aggregation guarantees anonymization and sorts out essentially the data privacy stuff. Consent. Now, a lot of people say you have to have prospective consent for a study on a piece of tissue or some blood or a test, and a different consent for every time you use it again. There's already been an act about the human tissues which showed that this was unworkable, bureaucratic, and destroyed major infrastructures for healthcare. like cancer depositories. So it's already been changed. Okay? So people are going more and more towards, you give your tissue or whatever for research of any type.
That's called broad consent.
And if you're anonymized, you don't need any of this anyway. The laws of the land say, the laws of Europe say, if you give data and then those data are analysed with no way of getting back to you, you need neither consent nor anything else. And then the ethics, we have local ethics committees that vet experiments. They increase the value and credibility of the science. They need to see, is it a good question? Have you got enough data power-wise? Have you got the analytical tools in order to give you an answer? Is the answer going to be relevant? That is ethics. It's nothing to do with little doves in the air coming down onto your head, as in Italian painting of the 15th century. It has to do with whether you are using the human as an experimental object and whatever the human gives you in a way which is of use to the whole of humanity. So this is the solution. The solution is that all this in the under the red line remains under the hospital, as is. The only additional bit is this bit. So the hospitals keep control of their data, so we don't move the data, we don't corrupt the data. So the IT guy in the hospital is happy. The IT guy in the hospital produces archives, off-site usually, just in case something gets corrupted with primary data. We're asking for one archive, so we need 20 to 50,000 Swiss francs to put in infrastructure to pick up this archive, during which we will uniformize the files in real time and depersonalize them with industry standard depersonalization, taking off the headers which give you names and birth dates and stuff like that. We will, on those data now, pre-process them, denoise them, that means smooth, remember from your imaging, standardize, 0, plus, -1, normalize anatomically, John Ashburner, numerical normalization, and then we will prepare them for query, for mining all the data, and for specific questions selected data, which will be identified by the distributed query mechanism, which Cesar and Anastasia Alamarki and Manos have been creating over the last year and a half. That works. We know it works because we do it every day anyway. But here, in a highly specific situation, and actually on what are essentially the raw data files, slightly pre-processed. Then we will aggregate that information which is necessary for the question, or all of it, encrypt it, filter it on the output side for depersonalization and anonymization, and send it to the inside portal, where it will be added to all the other end databases doing exactly the same thing. And it will give you then an aggregate of aggregates where you'll be able to graph it, you'll be able to do whatever you like with it. That will give you the result, either a constant refinement of the disease signatures within the brain disease space. It will allow you to visualize it, allow you to analyze it. And when you've done that, with the system that's been put in place by our colleagues, you will remove all these aggregates. Don't keep them. Let's keep it simple. All you'll keep is the provenance of each piece of data as it came through in order to give you your result, so that you can exactly reproduce your result if necessary. So it's storing the processing history and the data is deleted. That's the strategy, and we've got most of it in place now. So that we're hoping that at the end of this month we'll have the first prototype will be issued to the Human Brain Project. And people will begin to play. And Ferrat and his people have been playing with French data, a lot of French data, just given for this purpose. Scans, clinical scales, measurements, genes, proteins, CSF, some of it incomplete, some of it complete, 500,000, 1 million SNPs and so on. Integrating it, just using Excel, but you need, it's better to have something slightly more sophisticated in the long run. And finding things like this, which are groups of diseased people That's Alzheimer's, it must be, it's associated with A-beta. Normal people in normal people in blue. Different groups of normal people, perhaps some with preclinical, we don't know yet, we have to follow them up now. Some perhaps normal, some perhaps early aging, some perhaps late aging, with different haplotypes, that's the black things. Different PET data, the yellow spots. Different MRI data showing patterns of atrophy. That is the Alzheimer pattern of atrophy, the biggest one with disabling cognitive function. So I think that, and then there are lots of ways to visualize this. This is, I think yours, is this yours, Manos? Oh no, sorry, that's the wrong one. Why didn't it start? Sorry, I must show you this, it's so pretty. This is my final slide. So put them in, I think. Let's go back, forward, everything. Go forward. Go forward. Go. No, it's not going to work. Is it working? Yeah. Isn't that pretty? So these, the red things are like the rules, which compose of haplotypes and patterns of atrophy. Look how some of them form little islets which seem divorced from everything else. Look at that one. Okay. The different haplotypes, so a PET pattern which is hippocampal there, but other ones are linked together. So it begins to really fill out that disease space, and each set of rules, of course, gives you an idea about targets for treatment. They may be genetic, they may be other. It will depend on which particular sets of abnormalities were found. Okay, so that really gives you the sort of the clinical background, and I've finished, and I'm happy to discuss. Thank you.